Hi, everyone, and welcome to OR Today webinars. We have a great presentation for you today featuring Anastasia Johnson, Clinical Education Consultant of Advanced Sterilization Products. Stacy will cover patient safety related to current endoscope reprocessing methods. Attendees will learn about the FDA Safety Communications released in 2020 and 2021 related to endoscope reprocessing and how to apply the evidence to plan for reprocessing endoscopes in the future. Stacy Johnson is a clinical education consultant for advanced sterilization products, providing clinical support and training for high-level disinfection and sterilization systems. She worked as an RN for 18 years in surgery, with 10 of those years as a clinical nurse specialist. As a CNS leader, she supported evidence-based surgical and procedural patient care and OR safety. Stacy currently serves as the AORN Twin Cities Chapter Board. OR Today would like to thank our sponsor, Advanced Sterilization Products. Every day, millions of people around the world visit a healthcare facility. The care and handling of instruments used to treat patients is vital to their health outcome, and there is no room for error. ASP is a leader in infection prevention, dedicated to creating the products, solutions, and processes needed by practitioners to protect patients during their most critical moments. We are committed to continuously advancing infection prevention technologies that healthcare depends upon. Above all, ASP puts patients first. Please visit asp.com for more information. For nearly 20 years, OR Today has provided perioperative and SPD professionals with up-to-date news and information about their profession, including new guidelines, techniques, and equipment, as well as practical information for career building, problem solving, and overall well-being. Please visit ortoday.com to subscribe to our newsletter and magazine. Today's webinar is eligible for one continuing education hour by the State of California Board of Registered Nursing. You can obtain your certificate by completing today's post-webinar survey. More details on this at the end of today's webinar. Should you have any questions during today's presentation, please submit them at any time during the webinar using the questions feature on the GoToWebinar dashboard. As I mentioned earlier, our presenter today is Stacy Johnson. Stacy, you may begin whenever you are ready.
Hello. Today we are presenting FDA Safety Communications, Implications for the Future of Reprocessing Flexible Endoscopes. We have three objectives today. 1, discuss patient safety related to current endoscope reprocessing methods. 2, discuss FDA safety communications released in 2020 and 2021 related to endoscope reprocessing. And finally, we're going to apply the evidence to plan for reprocessing endoscopes in the future. Today, what we do know is that millions of endoscopy procedures are performed annually in the United States, and endoscopes are increasingly complex in design and difficult to clean. The guidelines governing the cleaning and disinfecting of endoscopes, which include colonoscopes, gastroscopes, bronchoscopes, duodenoscopes, etc., so that they can be used again, are over 50 years old. Even after being cleaned and disinfected, studies show a significant number of endoscopes remain contaminated with potential pathogens. Over the past 10 years, evidence has mounted that endoscope reprocessing is frequently not effective. Reprocessing failures can have dire consequences for patients, and infections have been associated not just with duodenoscopes, but with ureteroscopes, cystoscopes, and bronchoscopes, colonoscopes, gastroscopes. As the complexity of surgical procedures has increased, the complexity of instrumentation has also increased, making it more challenging to effectively clean and reprocess these devices. In 2015, patient infections related to contaminated duodenoscopes used in ERCP procedures made the headlines. For those of you in clinical practice in 2015, you may remember these unfortunate patient safety events and the laser focus that was placed on endoscope reprocessing after this time. So this isn't new news, but what's happening today? In 2021, endoscope infection risks are still in the news. According to Dr. William Rutala, semi-critical items represent the greatest risk of disease transmission because far more healthcare-associated infections have been caused by reusable semi-critical items than critical or non-critical items. Many of you participating today are familiar with the Spalding classification system, so we will just briefly review and give us short history. In 1968, Earl H. Spalding developed a rational approach to disinfection and sterilization of patient care items and equipment. Spalding believed the nature of disinfection could be understood readily if instruments and items for patient care were categorized as critical, semi-critical, and non-critical according to the degree of risk for infection involved in use of the items. Items defined as critical are those that enter the sterile tissue or the vascular system or through which blood flows and require cleaning followed by sterilization. Semi-critical instruments are defined as any device that contacts intact mucous membranes. The recommended reprocessing guideline for semi-critical objects is cleaning, followed by high-level disinfection. Non-critical items come into contact with intact skin. The current challenge is the heavy microbial load that may be present on some flexible endoscopes and the difficulty in cleaning and disinfecting long, narrow channels. Current HLD or high-level disinfection processes may not be adequate to ensure flexible endoscopes are safe to use on patients. Dr. Rutala has proposed modification of the Spalding system, wherein items secondarily entering sterile tissue or the vascular system are considered to be critical items. Devices secondarily entering sterile tissue would include devices that enter by way of mucous membrane, such as a bronchoscope, a cystoscope, or a duodenoscope. For example, a bronchoscope enters the bronchi through the mouth or nose, a cystoscope enters the bladder through the urethra, and a duodenoscope enters the bile ducts or pancreatic ducts through the mouth. Requiring sterilization of items that secondarily enter sterile tissue or the vascular system may help prevent patient infection. In 2021, the Multi-Society Task Force on Reprocessing Flexible GI Endoscopes and Accessories acknowledged these considerations and has determined that given the environment in which endoscopes are introduced and used is not sterile, and thus each time that the endoscope enters the digestive tract, sterility is immediately broken. Reclassification of endoscopes within Spalding is not advocated. So officially, Spalding classification is not changing. Yet, it's still important to consider how items that are entering sterile tissue are being reprocessed in healthcare organizations. We will now review the important distinguishing characteristics of sterilization versus disinfection. Sterilization and disinfection are necessary to ensure the safe reuse of medical devices. Sterilization is a process that destroys all forms of microbial life. And high-level disinfection eliminates many or all pathogenic or microorganisms, except bacterial spores. A picture is worth 1,000 words. This graphic representation demonstrates a simulated microscopic view of how a contaminated item, once cleaned, reduces the microbial load. Disinfection reduces all microbes except spores, and sterilization eliminates all microorganisms. According to the Association for the Advancement of Medical Instrumentation, AMI, as we know it as the Bible or the go-to gold standard for reprocessing items, SD91, which focuses on flexible and semi-rigid endoscope reprocessing in healthcare facilities, notes that high-level disinfection is frequently used to reprocess endoscopes. Flexible endoscopes are cleaned at the point of use immediately after each procedure, followed by manual cleaning and high-level disinfecting using chemical disinfectants. At minimum, it is recommended that devices are subjected to HLD after each use. But when possible and practical, flexible and semi-rigid endoscopes should be sterilized due to the greater margin of safety built into sterilization. Notably, when Dr. Spalding developed his classification system, some of the pathogenic organisms that put patients at risk today were not included. High-level disinfection is a multi-step process and is expected to be able to inactivate most pathogenic bacteria and viruses and fungi, but may not reliably inactivate certain types of microorganisms, including bacterial spores. When these devices are used in sterile tissue procedures, sterilization is recommended. Now let's look at some research studies that have been looking at endoscope reprocessing. In 2017, Dr. Corey Ofsted and Associates conducted a study noting that, starting with the fact that patient injuries and infections had been linked to ureteroscopes, but there were no guidelines or quality benchmarks specific to ureteroscope reprocessing. 16 ureteroscopes at 2 sites were examined after manual cleaning and sterilization. So these scopes were ready for patient use. All 16 demonstrated visual irregularities, which included discoloration, residual fluid, foamy white residue, scratches, and debris in the channels. All 16 scopes were tested to detect contamination levels, and they exceeded the national benchmarks which have been established for GI endoscopes. The researchers concluded that flexible ureteroscope reprocessing methods were insufficient and may have introduced contamination. They noted that no bedside point of use pre-cleaning was performed by OR personnel. No protocol was in place to report delays between procedure and the initiation of manual cleaning. And no alcohol flush or sufficient forced air exposure was performed to ensure drying before sterilization. Now, the OR managers stated these steps were performed by OR staff, but the observers did not see them in action, and OR staff acknowledged they did not routinely practice these. Findings of this study reinforce the need for frequent audits of reprocessing practices and the use of cleaning verification tests and visual inspection as recommended in reprocessing guidelines. So what is happening in your departments, in patient care areas where we're reprocessing endoscopes? You might know your practice appears to be stellar in relationship to endoscope reprocessing. Dr. Ofsted and Quick and Associates studied in 2018 a multi-site assessment. where they found bronchoscope high-level disinfection reprocessing methods were ineffective, even when performed in accordance with endoscope reprocessing guidelines. To reduce patient exposure to contaminated bronchoscopes, the researchers recommended institutions should consider shifting from high-level disinfection to sterilization. Key organizations such as AMI and the Association of Perioperative Registered Nurses, AORN, are calling for a shift to sterilization. If you can sterilize an item, you should. We will now discuss the FDA Safety Communications regarding endoscope reprocessing. The U.S. Food and Drug Administration, or FDA, takes the risk of patient infection very seriously, and they have released 3 recent announcements regarding safety precautions related to endoscope reprocessing, which implies a shift of reprocessing standards. For those of you in busy clinical practice, hoping that this will give you some insight into these recent communications. The FDA convened scientific and clinical experts in a meeting in November of 2019. The panel convened to discuss the technological design advancements and effective reprocessing of duodenoscopes that will enhance the safety of these devices. The FDA encourages the public to report problems with medical devices through the MedWatch voluntary reporting form. This includes patient infections or device contaminations associated with endoscopes. Healthcare personnel employed by facilities that are subject to the FDA's user facility reporting requirements should follow the reporting procedures established by their facilities. The information collected from medical device reporting, or MDRs, between 2015 and 2019 reveals there were declines in reported infections and patient exposures. However, reports of contaminated duodenoscopes rose during this period. Though the risk of infection from inadequate reprocessing is relatively low, an ERCP and endoscopy are necessary and life-saving procedures, so the continued availability of duodenoscopes to perform these procedures remains a critical public health need. In July of 2020, the FDA put out a safety communication updating their 2019 recommendation of transitioning to duodenoscopes with innovative designs to enhance safety. Duodenoscopes play a vital role in the assessment and treatment of diseases and conditions of the pancreas and bile ducts. They are used in more than 500,000 ERCP procedures each year in the United States. These devices have complex designs that include reusable, hard-to-clean components. Fixed end cap duodenoscopes have a plastic or rubber cap permanently glued to the metal edges around the distal end to prevent tissue injury from the metal edges of the scopes. When permanently affixed, the end caps also limit the accessibility to clean the crevices at the distal end. The FDA recommendations FDA has asked each duodenoscope manufacturer to transition away from fixed end cap duodenoscopes to those with more modern design features that facilitate or eliminate the need for reprocessing. Hospitals and endoscopy facilities should transition to innovative duodenoscope designs that include disposable components such as disposable end caps or to fully disposable duodenoscopes when they become available. Recommend healthcare facilities purchase new duodenoscopes or begin to develop a transition plan and work to replace their conventional duodenoscopes with newer models. What they're saying is that for those practices that are reprocessing endoscopes, focus carefully on innovative models that are coming forward, pay close attention, keep in close contact with endoscope manufacturers and know what is what is currently coming forward for use with patients. Stay on top of the new technology and make recommendations to your organization for repurchase and make those as they become available. Failure to correctly reprocess a duodenoscope could result in tissue or fluid from one patient remaining in a duodenoscope when it is used on a subsequent patient. And in rare cases, this can lead to patient-to-patient disease transmission. As always, ensure staff are appropriately trained and are meticulously following reprocessing instructions. Also, institute a quality program that includes sampling and microbiological culturing and other monitoring methods. These can include, beyond culturing, there are ATP tests on the market, tests that test cleaning processes. This is a time to work with multi-stakeholders within an organization, infection prevention, laboratory, and figure out what kind of quality control program you could implement in your facility. Develop schedules for routine inspection and periodic maintenance in accordance with duodenoscope manufacturer's instructions. And consider reprocessing with supplemental measures such as sterilization or use of liquid chemical sterilant processing system consistent with the device's labeling. In April of 2021, the FDA released a report announcing their investigation into reports of infections associated with reprocessed urological endoscopes. The investigation includes numerous MDRs describing patient infections and other possible contamination issues associated with reprocessing urological endoscopes. This includes cystoscopes, ureteroscopes, and cystourethropes, devices used to view and access the urinary tract. From January 1st, 2017 through February 20th, 2021, the FDA received over 450 MDRs describing post-procedure patient infections or other possible contamination issues associated with reprocessing these devices. Outside of the US, 3 reported deaths were associated with these infections and contamination issues from reprocessed urological endoscopes. The FDA is emphasizing the importance of following the manufacturer's labeling and reprocessing instructions for these devices, including accessory components for cleaning and subsequent processing to minimize the risk of infection. Reprocessing includes either pre-cleaning, leak testing, cleaning, disinfecting, rinsing, and drying, or in other words, high-level disinfection, or pre-cleaning, leak testing, cleaning, and sterilization. Are you following all these steps in your 4 urological endoscopes? Is the process complete from the procedural area or the operating room through the reprocessing area? In June 2021, the FDA released a safety communication on updated reprocessing recommendations for flexible bronchoscopes. In March of 2015, the FDA provided a guidance document. And in that document, bronchoscopes were identified as being part of a subset of devices that pose a greater likelihood of microbial transmission and represent a high risk of infection if they are not adequately reprocessed. A new recommendation from the FDA is to consider using single-use bronchoscopes in situations where there is an increased risk of spreading infection. These examples are when multi-drug resistant microorganisms are present, in immunocompromised patients, or patients with prion disease, or when there is no support for immediate reprocessing of the bronchoscope. This can be common in a large organization where Life-saving procedures are occurring at different parts of the hospital, and it's difficult for those endoscopes to get back to the reprocessing area of the hospital in a timely fashion. When treating patients with coronavirus COVID-19, refer to recommendations from the American Association for Bronchology and Interventional Pulmonology. As reported in 2015, between January 2010 and June 2015, the FDA received 109 MDRs related to infections or device contamination associated with reusable flexible bronchoscopes. Since that time, the FDA received 876 additional MDRs between January of 2015 and January of 2021. It's related to infections or device contamination associated with reusable flexible bronchoscopes. Of these, there were 7 reports of deaths. Factors contributing to infections included failure to follow manufacturer instructions or continued use despite device integrity, maintenance, and mechanical issues. Since 2015, the number of MDRs relevant to infection or contamination submitted to the FDA has increased from under 100 per year to between 100 to 200 per year. There are approximately 500,000 bronchoscopy procedures performed annually in the United States alone, and the MDRs described here were received from all over the world. For reusable bronchoscopes, consider using sterilization instead of high-level disinfection when feasible, because sterilization has a greater margin of safety than high-level disinfection. Steps should include the pre-cleaning, leak testing, cleaning, and sterilization. If sterilization is not available, then high-level disinfection should include pre-cleaning, leak testing, cleaning, high-level disinfecting, rinsing with tap or utility water followed by alcohol flushing or with critical filtered or sterile water and drying. And use only manufacturer-specified cleaning accessories, high-level disinfectants, enzymatic cleaning agents, and detergents. For those of you working with the directly handling the reprocessing, find out if you are using the appropriate manufacturer-specified cleaning accessories and cleaning agents. The FDA also notes this about the MDR system we have referenced today. Although the MDR system is a valuable source of information, it is a passive surveillance system and voluntary reporting. It should not be used to calculate the incidence of a specific event. So it's not often possible to definitively determine the cause of the event based on the information. MDRs are not by themselves definitive evidence of a faulty or defective medical device, and they cannot be used to establish or compare rates of event occurrences. Given overwhelming evidence of potential points of failure in endoscope reprocessing via traditional high-level disinfection, what can be sterilized should be sterilized. With the infection risk that endoscopes present to the patient, terminal sterilization is the preferred method of microbial inactivation and the only option in sterile environments. Terminal sterilization is recommended for flexible and semi-rigid endoscopes that enter sterile body cavities. Terminal sterilization is required for all endoscope accessories that penetrate mucosa, such as biopsy forceps and sphincterotomes. What cannot be sterilized should be reprocessed in an advanced endoscope reprocessor, which offers an advantage over manual reprocessing. They automate and standardize several important reprocessing steps and reduce the likelihood that an essential reprocessing step will be skipped. An automated process for cleaning and disinfection may be more efficient than manual processing. It may also result in less user exposure to toxic chemicals and help ensure repeatable results. This aligns with FDA recommendations that users should be instructed to thoroughly clean semi-critical devices and then reprocess them by sterilization. If the device design does not permit sterilization, For example, the device materials cannot withstand sterilization, then high-level disinfection should be used. Thank you for participating today. We will go to a question and answer session.
Thank you, Stacy, for a great presentation. We do have some questions that have come in. Our first question is, some hospitals and ASCs believe the costs of HLD or sterilization for endoscope reprocessing outweigh the risk reduction benefits. Is there evidence that the reverse is true, that the benefits outweigh the costs?
Well, there certainly are costs associated with, you can even say hospital-acquired infections, statistics on those, and certainly can be looked up if someone wants the exact statistics from CDC and other sources that would list that. But really, when you're talking about what we had discussed today in the FDA Safety Communications that have been provided to inform the public and healthcare providers of safety risks and the recommendations to mitigate those risks for reprocessing endoscopes. In the articles we referred to, healthcare institutions have been in the news for contaminated endoscopes have been associated with patient infections and patient deaths. Preventing negative publicity and preventing loss of confidence in healthcare are certainly benefits. And most importantly, preventing human suffering are just some of the costs that we, to not prioritizing the safest practices for endoscope reprocessing. And not wanting to forget not only the unfortunate incidents with patients and their families, but also the people who are in healthcare who are caring for patients and trying to do their utmost best. We need organizations who are in the leaders to see the benefit in investing in improving endoscope reprocessing.
Thank you. Our next question is, when is the best time to measure ATP on endoscopes? For example, should you do an ATP test right after the endoscope has gone through HLD or after drying and hanging it in the cupboard?
This is a great question, and a lot of organizations and people who are involved with endoscope reprocessing are asking these questions, and infection preventionists. AMI recommends that cleaning verification of flexible and semi-rigid endoscopes should include visual inspection combined with other verification methods. So, this visual inspection and including a true visual inspection in your processes is important. And, that's to try to assess both external surfaces and internal housing and channels. So working in multidisciplinary teams in an organization and looking at policies and procedures and trying to determine what the best practices would be to institute those kinds of tests and using instrumentation like a borescope is certainly important. But as far as cleaning verification tests, common markers include protein, carbohydrate, hemoglobin or blood, and ATP. So those are cleaning verification tests and kits that are available on the market. Again, real-time testing of endoscopes should be done, as the question asks, prior to high-level disinfection. So immediately after manual cleaning and then prior to high-level disinfection. And the reason is that so that you can re-clean anything that you see that's not meeting your standards. And that is recommended by SGNA, Society for Gastrointestinal Nurses and Associates. Thank you for the question.
All right, our next question is, do you see endoscope reprocessing moving from requiring high-level disinfection to sterilization?
Well, we've talked today about professional guidelines that instruct users to sterilize semi-critical devices whenever possible. And that's both AAMI and AORN, sterilize when you can. But Dr. Rutal and Dr. Weber have published extensively on the topic of endoscope reprocessing and the challenges that we face. And they discuss, that the FDA, as part of what they are reviewing, is that this is really an industry issue where the manufacturers of endoscopes, of reprocessing equipment, high-level disinfectants, sterilization technologies, along with our federal authorities, CDC, FDA, National Institutes of Health, everybody needs to be engaged in providing adequate resources to design and complete studies for moving forward for improved reprocessing technologies for endoscopes. And I think this focus of the FDA Safety Communications is really to get organizations to really pay attention as things are changing and evolving in the industry, so that you're planning to make changes, maybe starting to implement some of these quality measures and quality testing measures, and be ready to adapt to anything new that comes forward that is going to improve reprocessing in the future. Thank you.
Thank you so much, Stacy, for your time today and for a great presentation. I'd like to encourage everyone to visit our sponsor to learn more about the products and services they provide to our industry. Please visit asp.com. A quick reminder that you can obtain your certificate by completing today's post-webinar survey. The survey will be sent out to you 1 hour after the completion of today's webinar. You must complete the survey to receive your 1 continuing education hour, and you'll be able to download the certificate once the survey is submitted. If you have any questions, you can reach us at webinar@mdpublishing.com. We'll be back soon with another webinar. Please visit ortodaywebinars.live for more details and complimentary registration. Thank you all and have a great day.